So thanks for inviting me to your really interesting set of talks. It's been a lot of stuff I didn't know the details of the history, and it's really fascinating. So in my talk, I'm replacing Anna Lindholm, who was going to do it but had to drop out. So it's a bit of a rush to put this together. Also, I am recovering from COVID, so I may be stupider than usual. So I do apologize for that. Um, Now, what I want to talk about is one of Mendel's the kind of stereotypical view that we've just been told is a terrible idea of what Mendel discovered and what he did. One of the rules he discovered is that segregation is fair. That whether you're talking about genes, a single gene that causes everything, or whether you're talking about traits, it's fair segregation. And I'm going to be talking about the work since Mendel, some of it very early on in the 20th century, that shows that that's not always the case. So here are some of my key collaborators. I'll be introducing them more as we go through or crediting them. So yeah, fair segregation is one of the key things that Mendel suggested was going to happen because that's what he saw in his pea plants. And what he was really talking about there, although he didn't know it at the time, was these were genes that were being controlled by, these were traits that were being controlled by nuclear genes. And so I'm not going to be talking about endosymbionts or anything that gets passed on through cytoplasm that only get transferred through eggs. And I'm also not going to be talking about any parasitic genes or chromosomal drive B chromosomes, things like that, because they're too complicated. They do my head in. But there's a fundamental conflict. When you have a diploid organism that has two copies of every chromosome, and it produces gametes that each only carry one copy of a chromosome. If you are the blue chromosome in this picture, and you can somehow disable the yellow chromosome sperm, then all the offspring of that male will carry that blue chromosome. So that's a huge fitness advantage. Whereas the yellow chromosome, if that only gets carried on in a normal male, that only gets onto 50% of the offspring, it's much less fit. So every gene on every chromosome has this potential ability to try and increase its percentage of representation in the gametes or representation in the offspring. So it's not just genes competing by making better individuals and trying to be a more successful mouse. It's also, can you be a more successful sperm within that mouse? So one of the first people to discover these kind of things was Nadine Dropovalskaya-Zavadskaya, who was an amazing woman. You should read up about her. She was a Russian surgeon who also did a lot of work on mice. She discovered this thing called the T haplotype. And it's an autosome, one of the mouse autosomes, I think maybe the 23rd chromosome. It is something like 2 million years old. So this gene has been, this haplotype has been around for a very long time. And most of this chromosome is taken up by an entire inversion. So for anyone who doesn't know, when chromosomes link up during meiosis, when you're creating the divisions to create a single haploid egg or sperm, all the chromosomes line up together next to each other in their pairs. And this allows crossover. So genes can jump, alleles from one chromosome can jump to replace the allele on the other one. It creates recombination, which allows generation of novelty in the offspring. But sometimes a section of chromosome will flip over and recombine. So within its chromosome, it's just flipped over. And you can see that here in the diagram, where these sections of chromosome flipped. And then at that point, they know that the genes are no longer next to the opposite, the gene on the same gene on the other chromosome, and you can't get crossover. So effectively, this whole recombined area is inherited as a single block. And that seems to be very closely associated with these kind of selfish drive in chromosomal drive systems. So in this mouse, if a male carries the T haplotype on one of his chromosomes, that will be passed on to about 90% of his offspring. The normal chromosome only gets into about 10% of the offspring, and that's because the T chromosome produces a toxin when the sperm are being formed, which gets into all of the sperm because they have cytoplasmic bridges, they're all connected. And it also produces an antidote to that toxin, and that antidote is only produced in the sperm that carry the T chromosome. So it poisons everyone and then takes a pill to save its own life. And that means that the non-T sperm, they're not killed, but they're damaged to the point where they can barely swim. So that's sperm drive. You can also get drive in through the female, through ova, both in mammals and plants. So Lila Fishman has done a lot of this work in monkey flowers, and Takeshi Akera has done some great work on this in mice. And so in the production of ova, so seeds and eggs, what actually happens is there's the 4 products of meiosis. There are these 2 splits. Chromosomes double in number, so you get 2 copies of every chromosome, and then they all split into individual single cells, each carrying 1 chromosome. But in female meiosis, 3 of those are destroyed, and only 1 of them becomes an actual egg. And so if you can avoid being in one of those polar bodies that get destroyed, you'll get into a disproportionate number of offspring. And so Takeshi did some really great work on this, showing that the centromeres, which are these big areas, often in the middle of chromosomes, that don't code for any genes. They're just repetitive, highly repetitive, random noise, but they're really important binding sites that the chromatin, which is kind of scaffolding inside the cell that moves chromosomes around during meiosis, they bind very strongly to the centromeres and pull centromeres in different directions. And so centromeres that are particularly big, he's discovered, are much more attractive, and they tend to get pulled into the middle of the cell, which forms the egg, while smaller centromeres get pulled to the side and ejected as a polar body and then destroyed. And this is kind of drive is not just found in mice, it's also found in humans. It's very rare. So sometimes humans will have chromosomes that fuse together. So 2 chromosomes will link via their centromeres, and that will create a massive centromere that's twice as big as a normal one. And that centromere is very, very good at staying in the egg and not being destroyed in the polar bodies. And the kind of drive I work on is X chromosome drive. So sex chromosomes are very different. So it's very easy for a sex chromosome to find something to poison on the rival sex chromosome. And that will massively increase its success. So in this fruit fly Drosophila simulans, if in normal males you have a Y chromosome and an X chromosome, and half the sperm carry the Y, half the sperm carry the X, half of the offspring are sons, half of them are daughters. But if the X chromosome can somehow kill the Y chromosome sperm, then all of the offspring will carry the X, and that X chromosome is passed on to 100% of kids. And so that is an absolutely massive selective advantage. So in studies in nature, a selective advantage of 1% increase over your rival alleles is considered a strong selection. And a 5% advantage would be absolutely huge. What we're talking about here is 100% advantage over rival alleles. So this is incredibly strong selection. They can spread very rapidly through populations. And of course, the other thing you can obviously notice about this is if you get to fixation in a population, if 100% of the males carry this driving X chromosome, in the next generation, everything will be female and they'll go extinct. And I've done some lab work where I've shown that under some circumstances, you can make that happen in labs where I lost populations within 9 generations because everything became female. And these things can spread incredibly rapidly. So some brilliant work by Catherine Montchamp-Moreau and her team in Paris found this drive system in simulans in the Mayotte Islands, in just off the coast of Africa in the year 2000. By 2010, this gene had spread through the populations up into Egypt and into Israel. By 2016-17, it spread right the way around North Africa, into Morocco, and into West Africa. So that's an incredible rate of spread. Now, to some extent, it's because simulans is probably riding around in trucks full of vegetables. But even so, it's an amazing spread. There are no real invasive species I can think of that can spread as fast as this driving gene has managed to do it. And so these gene drives are found in all sorts of different organisms, including major pests like mosquitoes that carry major diseases, tsetse flies, agricultural pests like medflies, mice, other rodents, and also fungi, plants, just about everything where you have this fundamental conflict where you have diploid chromosomes being divided into haploid gametes. And so there's this idea arose that, well, this should be happening everywhere. And so we should see loads and loads of drive all the time. Mendel was wrong. Loads of alleles will be constantly battling to drive better than rival alleles, and so Mendelian inheritance being fair is just incorrect, and so gene drive should be really common. And Mendel was wrong. But no, that's not true. It turns out Mendel was broadly correct. Most genes don't cheat, as far as we can tell. So the best data for this comes from humans, where people have sequenced individual sperm from men and sequenced the man, and then found that there's almost no deviation in the frequency of different alleles. It just seems to be completely fair. There's weaker evidence, but we can look for genetic sweeps in loads of different animals that have been genome sequenced. And again, we just don't see very much of this stuff. So the people who are into meiotic drive appear to have been wrong about this. The ones that we do see, we often find in model organisms, and that suggests that these things are quite rare, and it's only when you really study a species an awful lot, there are loads of biologists working on it, that you find it. They're often rare within the species, so they're only found in some populations, or they're not at high frequency. Some of them, like the mouse T haplotype, are old, in some cases older than the species that carries them, and a lot of them have severe costs. So some of my work, well, I say my work, this has actually included data from Theodosis Dobzhansky, who was one of the real leaders of the modern synthesis in the sort of 1920s, 30s, 40s. So this is Drosophila pseudoobscura. It lives across North America it lives all the way down into Colombia, and it carries this driving X chromosome. And so the orange parts of the circles are the proportion of proportion of chromosomes in this population that are a driving X chromosome compared to the non-driving X chromosome. And you can see it's commoner in the south than in the north, and it has been from all the data we have for the last 80 years. So from Dobzhansky wandering around on a horse, catching fruit flies in his amazing work in the 30s and 40s and 50s, to my most recent collections in Arizona and Utah and Montana. And in this case, what seems to maintain this decline is that female fruit flies in the north almost always mate with multiple males. And so within their uterus, the sperm from multiple males will fight it out, try to get to the egg. And males that have drive, half their sperm are dead. Even their functional sperm are slightly damaged by this killing process. And so they can't compete with the normal male sperm. Whereas in the south, about half of females that I find in the wild have only mated with one male, and that's really good for the drive males. So yeah, we get geographic differences. We also showed that you could get the rapid evolution of increased mating by females. So females went from mating with males every 3 to 3.5 days to mating with males every 2.5 days. Within 12 generations, we putting them in lab populations in the lab. Basically, if there was drive in the population, the females responded by evolving increased mating with males, because they increased the chance they'd mate with a male who had normal sperm, who could produce Y chromosome sperm, and therefore they would be able to produce sons, which massively increased their fitness. They weren't doing it on purpose, of course. It was just that was the evolutionary process. And so, because we haven't published this yet, but we have some data that should be hopefully coming out next year showing that the genes that control female remating frequency are spread all over the genome. There's loads of them. It's genomes everywhere. This is a real parliament of the genes controlling selfish genes situation, which was an idea proposed by Egbert Lee back in the 60s, I think. So there's loads of the genes that have no interest in this drive are basically slamming down on these driving chromosomes, stopping them from being able to spread. And some modeling paper by Stu West and his PhD student Tom Scott, I think, did some nice modeling to suggest that this should generally be pretty common. So was Mendel wrong? Well, no. I see it a lot like a shopping trolley with a badly damaged wheel. It kind of, a lot of the times it goes in the direction it's supposed to go in, but it wobbles all over the place. There's a lot of conflict. There's lots of mild deviations, but then the rest of the genome shuts down these deviations. And so transmission is mostly fair. But every now and then it goes completely off kilter, and potentially populations or species die out because everything becomes male or female, or everything gets one particular variant of the chromosome. Why does this happen? We don't know exactly. I mean, it might be that suppression evolves very, very rapidly. I also think it's possible that there's effectively a kind of immune system, a genetic immune system that suppresses selfish genes in much the same way that humans have an immune system that is very generalized as well as specific immune function against targets we've experienced. Although that's controversial, I don't know if that's true. Another possible idea that I think is very interesting is that these kind of selective, the forces involved, selective forces involved here are so strong. It's one of the very rare areas where you might get species level selection or population level selection, where any species which allows Y chromosome drive might go extinct so quickly that species that have generalized mechanisms to prevent this might be more successful. But that's very controversial. And we don't really know enough about the evolution of new drives because we've never seen one. So we don't know if new drives are generally pretty bad at driving and have lots of costs. But the evidence for all of this is a bit weak, so take everything I'm saying with a massive pinch of salt. And if I was going to summarize the field, I'd say it's basically a series of long, convoluted anecdotes, potentially told by a drunk man in a pub. We know a lot about the few systems that have been really well studied, and we think there's loads of stuff out there, but we don't know about it. We don't know about weak drives. We don't know what happens to drives that have been long-term suppressed. There's a load more we need to know, and maybe we need some Darwin or Mendel or lone genius that doesn't exist to come in and sort it all out for us. So just before I stop, I just want to talk a bit about synthetic gene drives, because I know a lot of you are interested in the ethics of Mendel and Mendelism and his purposes, well, the results of his work. So obviously there are huge problems we need to solve in the world. So malaria mosquitoes kill hundreds of thousands of people a year and blight the lives of millions. There's pest species that are causing massive conservation crises. So, for example, New Zealand is attempting to eliminate 6 predator species by 2050 because they're having such devastating effect on the ecosystem of New Zealand. And we need novel techniques to do that, because traditional techniques like poisons have lots of negative side effects and they're just not that effective. So we need to deal with these things cheaply and effectively. And if you have this incredible transmission advantage that drive has, you might be able to use that to modify natural populations. And this is something that's happening right now. So the idea is that if you wanted to grow a type of mosquito that couldn't transmit malaria, you could probably do that in the lab. But if you released it, it would just die out because the mosquitoes in nature would defeat it completely, because nature is much better at building mosquitoes than we are. But instead, what you could do is you could perhaps create a package of genes that will make a mosquito unable to transmit malaria. Put that into a gene drive you've built, and then the gene drive only has to beat the mosquito's genome. It doesn't have to beat nature. And that seems to be possible. In fact, there are people who have already done this, and they're currently doing cage trials. They've done cage trials it's largely based in Imperial College. They've done cage trials there, and it works. They're now doing cage trials in, I think, Benin in West Africa, with the possibility they will actually deploy these sometime soon. Where the idea is you build a Y chromosome drive that kills all female offspring, so all the mosquitoes turn male in an area. But also, again, if you have this very strong drive, there's huge ethical concerns. If Benin, say, decided to use this against their mosquito populations, what's to stop it spreading into Nigeria? Not really anything. If it goes wrong, who's to blame? Is it the government that allowed it? Is it the company or the researchers who built it? There's huge ethical considerations here. What about, there's the ethical question of, if you're a lab tech working on this, or a idealistic PhD student, and you have this gene drive and people are saying, no, we shouldn't release it. No, we shouldn't release it. You can make a moral argument. You should steal some of these and release it yourself, because that could save loads of lives. So I think these ethical questions are absolutely fascinating, and they're something we have to deal with, because these things are going to be able to be built. And another thing that's a risk is that you build one of these drive systems, this chromosomal drive system, which has the genes to make mosquitoes not transmit malaria. But someone else could then take that once it's been released, take that mosquito from the wild and then alter the package that it's carrying. So instead of making mosquitoes unable to transmit malaria, you can make them more likely to transmit malaria, perhaps. Or maybe if we're thinking about science fiction scenarios, you could give the mosquito a preference for the odor of a incense used in Catholic church services. So these mosquitoes can transmit malaria better, and they go to places where Catholics live. And so there's a kind of weaponization risk here as well, or a risk of bioterrorism. Now, in general, there's two ways to do it. You can either try and exterminate the species entirely, or you can try and modify the species. So this would be, for example, making the gut or the salivary glands of mosquitoes impossible to be infected by malaria or whatever the disease is. Both of those are possible. People are working on both of those things. Now, there's a very different version which uses an endosymbiont called Wolbachia, which was living in it, which are sometimes found in mosquitoes. But some researchers, just to see what would happen, took one from Drosophila melanogaster. They took this bacteria that lives inside cells and they put it into mosquitoes. And it somehow upregulated the mosquito immune system so that mosquito could no longer transmit dengue, Zika, chikungunya, and other diseases, viruses that are basically incurable. And so these mosquitoes have been deployed in northern Australia around Cairns. And after deployment, there were no more cases of dengue at all. It completely stopped it. It's also been very successful in Thailand, and it's been used in some other places. In some places, it's not so successful. Other places, it is successful. But these things are already being used. And this one's very successful. But the new gene drive ones, they haven't been used yet. I've been vaguely working on taking the T haplotype, which I was telling you about in mice, and putting the sex determination gene for mammals, the SRY gene, that if you have it, your body will switch from developing towards being a female towards developing as a male, putting that into the selfish gene. I didn't do that myself, but I did some modeling work with some people to try and work out how polyandry and promiscuity would affect it. And it is a risk. It makes it far harder to successfully deploy this. But the idea is you use this to wipe out mouse populations where they're invasive, where they were brought there by seafarers, and they're currently exterminating native species. So gene drives are a fascinating area. I think I'm out of time, so I'm going to stop there. But if you're a historian of science, it would be great if you could get a PhD student to start studying this, because this is history happening right now, and I think people in the future would really appreciate having a record of it. So thank you very much.